Hi everyone, my name is Suhelen Egan and I've come back to this forest to continue our discussion of the decomposition process. In the first module, we discussed what decomposition was, where it fits within the classical food chain, who the main decomposers are, and what their role they play is in the decomposition process. In this module, we're going to focus on one main learning outcome, and that is to show an understanding of the factors that influence decomposition. Just a short recap. Remember, decomposition is the breakdown of dead organic matter, or detritus, by physical and biological processes. When leaf litter hits the ground, one of the first things to happen will be the physical leaching of soluble minerals and organic compounds. Depending on the material, up to 30% of compounds in leaf litter can be dissolved in water. The remaining material consists mostly of complex carbohydrates, which will then be consumed by various detritivores that break down leaves into smaller and smaller subunits. This physical shredding of material helps to increase the available surface area and increases aeration for subsequent decomposition processes. The detritivores will assimilate only about 30 to 40% of the energy in the leaf litter, and the rest will either not be consumed or be deposited as waste. This is where decomposers, such as fungi and bacteria, become important. Through the production of specialized enzymes, they can break down more complex compounds that are recalcitrant to the detritivores. Let's take a closer look at some of those materials. In addition to simple sugars, plants, and in particular the cell wall components, contain some highly complex carbohydrate polymers that are difficult to degrade, including things like cellulose, hemicellulose, and lignin. As you might assume, the level of complexity of a carbohydrate molecule will impact its ability to decompose, with simple molecules such as starch having a faster rate of decomposition than more complex lignin or cutin. Cutin, by the way, is the waxy polymer you find on some plant material. The importance of leaf litter traits such as composition of plant tissue has been investigated in several studies. The one I'm showing you here was conducted in a freshwater stream in Germany. The investigators placed a predetermined weight of leaves from 9 different plant species into fine mesh bags and then deployed these bags into the stream. After 55 days, the bags were retrieved and weighed to calculate the amount of decomposition that had occurred and performed a regression analysis to show that the rate of decomposition of the leaf litter decreased with increasing lignin content. You can see the results here from this experiment in the graph. Each point here represents the results from a single plant species. This second study was conducted in a tropical rainforest and aimed to investigate how differences in carbohydrate composition can impact the time it takes for leaf matter from certain trees to decompose. The study used the same concept of mesh bags, but here they investigated the decomposition over a 250-day time frame. They then plotted the leaf mass remaining as a percentage on the y-axis in relation to the time in days on the x-axis. At the end of the experiment, 15% remains of the Brazilian fire tree compared to about 50% of the original biomass of the trumpet tree. On closer inspection, the researchers found that during the initial 50 days or so, there was a positive correlation between the solubility of the compounds that would leach out of each plant species and the rate of decomposition. That is, the more soluble the material, the faster it decomposed. Later in the experiment, they observed that the amount of lignin in each leaf type was negatively correlated with decomposition rate. So higher lignin, the slower the decomposition. This is a similar result to the previous leaf decomposition experiments we found in the German stream, suggesting some universal themes to the decomposition process of leaf litter. Specialization among microbial decomposers is thought to be one explanation for the differences in the rate of decomposition between different substrates. Let's take a look at the example of the potato that is predominantly composed of simple starch and a tree trunk that has high amounts of lignin. The theory is that the genes encoding for the enzymes to degrade the simple sugars, such as starch, as well as the generalist organisms that carry out this process, are widely spread and thus represent a larger range of taxonomically diverse microbes. In contrast, only a limited number of specialized microorganisms have the ability to degrade these complex polymers, such as lignin, found in our tree trunk here. Assuming this is the case, then you might expect to see a positive correlation between species diversity and traits related to decomposition, especially for organic matter comprised predominantly of simple substrates. For example, a group of scientists in Germany again investigated the dynamics of the microbial community during leaf litter experiment. They used mesh bags again and took samples at regular intervals to analyze key enzyme activities, microbial diversity, and physicochemical qualities of the leaf litter during a long-term decomposition experiment. One of the results they obtained is shown here in the graph. This correlation analysis provides evidence of a positive correlation between the number of bacterial species, also known as richness, and the activity of the simple sugar-degrading enzyme beta-glucosidase. That is, more bacterial diversity, the more enzyme activity they could detect. But the same study found a negative correlation with fungal species richness and the activity of enzymes that degrade more complex polymers such as lignin. This observation is in line with the theory that specialist decomposers are less diverse. If, for example, there are only a few fungal species in the leaf litter that can produce lactase, then the presence of other fungi may act to competitively exclude those specialists. So, if decomposition of lignin is the end goal, a diverse fungal community that are effectively will dilute the action of those lignin degraders and may not be actually beneficial. This was just one example. There are numerous studies that have tested hypotheses related to diversity and decomposition. To date, the most evidence suggests that diversity is important for decomposition, but the extent to which biodiversity impacts decomposition is context dependent. However, even with the right enzymes to break complex carbohydrates into smaller subunits, mineralization of the substrate is further limited by the chemical composition of the decomposers and the available resources. One of the main limitations to full mineralization of plant material is available nitrogen. The reason for this is relatively simple and comes down to the main differences in the structure of plants and animals. The major building blocks of plants are carbohydrates, and therefore they are carbon-rich. This is relative to animal cells that are predominantly made up of proteins and are therefore relatively nitrogen-rich in comparison. And it is this elemental stoichiometry that is reported as carbon-nitrogen or carbon-nitrogen phosphate ratios of the organic matter that determines the amount of nutrients released back into the environment. Let me try and explain this further with some examples. In this slide, you can see there is roughly 1 order of magnitude difference in the proportion of nitrogen between plant biomass at around 0.5 to 2% and that of bacteria or invertebrates averaging at about 10% nitrogen. In order to maintain growth, the invertebrates and microbial decomposers need to maintain an optimal carbon to nitrogen ratio of approximately 10 to 1. But, as we'll be discussing later module, some of the carbon is lost via respiration. For example, on average, bacteria will lose about 60% of the carbon they consume through respiration. Therefore, if we consider the carbon losses due to respiration, bacteria will need to consume an organic resource that is more like 25 carbon to 1 nitrogen. If, however, the ratio is higher, that is more carbon than nitrogen, all of the nitrogen is used up before all of the carbon is decomposed. If the ratio is less, that is more nitrogen, then some nitrogen will be released back into the soil. You can probably imagine how gaining a good understanding of the stoichiometric composition of different organic matter and the requirements for specific decomposers can be then applied to model optimal land usage and understanding global carbon budgets in response to climate change. So we've just talked about how chemical composition and characteristics like carbon to nitrogen ratio impacts the rate of decomposition. But what about other factors? Pause the video and take a moment to list at least 3 other factors you think might be important to facilitate decomposition. What did you come up with? If you are not sure, let's take a look at this graph showing changes in soil microbial respiration on their y-axis plotted against a function of soil moisture on the x-axis at 3 different temperatures. You might be thinking, why are we looking at how well microbes breathe? Well, remember, microbial metabolism is key to decomposition, so scientists will often use this as a proxy for decomposition processes. The first thing you might note here is that at higher temperatures, you have more decomposition occurring. But soil moisture has a sweet spot of around 40%, and it varies depending on the temperature. Moisture is not just required for metabolic processes, but many decomposers such as fungi, rotifers, and nematodes require a moisture film for motility, and therefore to physically access the organic resource. So we have things like temperature, water, nitrogen, and oxygen all having an impact on the decomposition process and success. The final thing here on my list is the physical structure or porosity of the organic matter. That is because the structure of the material directly impacts the level of aeration and available surface area for decomposers to access the substrate. One area where this is increasingly under investigation is the role of invertebrate animals as bioturbators of organic matter in aquatic sediments and soils. Here, the burrowing action of animals like mussels and worms create air pockets, bringing oxygen and acting as irrigation of deeper anoxic regions of the sediment, thus increasing the rate of decomposition. Bioturbation of aquatic sediments in mangroves and seagrass meadows, such as the photograph here shows, is considered an important environment for carbon sequestration and storage. Yet, there is still much we need to learn about the processes that impact the remineralization of the captured carbon. For example, in a recent study by Australian researchers, sediment microcosms were used to investigate the impact of the addition of organic matter in the form of seagrass with and without this bioturbator, a common marine yabby. The scientists' objective was to determine if the addition of seagrass as an organic matter source with and without the bioturbator could increase the rate of mineralization of carbon already trapped in the sediment. Let me walk you through one of their main results. Here, they have measured both oxygen uptake in the top two panels and total CO2 release in the bottom two panels in each of their treatment microcosms over a period of about 60 days. On the left of the treatment without the addition of seagrass, and on the right are the results from the treatment with the addition of seagrass to the microcosm. For each graph, you can see measurements for the treatments with the addition of the bioturbator in black and without in white labels. What I hope you have noticed is that there was a consistently higher amount of both oxygen uptake and carbon dioxide release from sediments that were bioturbated. While the authors of this work admit that this was an artificial setting, the results do highlight that the action of bioturbation could be a mechanism that converts seagrass sediment from what is classically considered a carbon sink to a carbon source, having important implications for climate change mitigation strategies. Well, that's all I have now for you. I hope this was useful, and remember that information in this module will be useful for you to gain ideas for your lab experiments on decomposition. See you next time.